Before the structure of DNA was recognized as a double helix, it was a problem of environmental control. In 1951, a single fiber of genetic material was a chaotic physical object, reacting unpredictably to the atmosphere around it. To see the molecule, one first had to dominate the air that touched it. Dr. Rosalind Franklin approached this not with speculation, but with engineering. She recognized that the clarity of an X-ray diffraction pattern depended entirely on the precision of a humidity chamber. While she could successfully calibrate the moisture levels inside a Philips micro camera, she could not calibrate the institutional flow of the data that resulted. Rosalind Elsie Franklin was born on July 25, 1920, into a prominent and politically active Anglo-Jewish family in Notting Hill, London. Her father, Ellis Franklin, was a liberal merchant banker who volunteered his evenings teaching electricity and magnetism at the London Working Men's College. Her family's commitment to community service was a central theme of her upbringing. They were actively involved in settling Jewish refugees from the Kindertransport during the lead-up to World War II. As a child, Franklin was noted for her intelligence and logical mind. Franklin was educated at Norland Place in St. Paul's Girls' School, one of the few institutions that taught chemistry and physics to girls at the time. By age 15, she had determined to become a scientist. She entered Newnham College, Cambridge, in 1938 to study physical chemistry within the natural sciences tripos. Although she completed her studies in 1941, Cambridge at that time did not award full degrees to women. She received only degrees titular, a formal status that was not retroactively changed until 1947. Her first professional work began in 1942 at the British Coal Utilisation Research Association, or BCURA. Coal was a vital strategic asset. It provided energy and acted as a filtration agent in gas masks. Her task was to understand why some carbons absorbed chemical agents more effectively than others. Franklin treated coal as a molecular sieve. She discovered that coal contains microscopic pores with fine constrictions at the nanometric scale. To measure these structures, she employed a methodology of rigorous precision. She measured the apparent density of various coals using liquids like methanol, water, and benzene. She then compared these to density measurements taken with helium gas. Helium molecules are small enough to penetrate the finest gaps. This allowed her to calculate the true density and reveal the internal architecture of the carbon. Her research demonstrated a clear relationship between heat and structure. As coal was heated to carbonizing temperatures between 600 and 1,000 degrees Celsius, its porosity increased. Simultaneously, its reactivity decreased. Substances were excluded from the pores in strict order of their molecular size. This work allowed for the first accurate classification of coals and the prediction of their performance. In 1946, she summarized her expertise with clinical directness. If ever you hear of anybody anxious for the services of a physical chemist who knows very little physical chemistry, but a lot about holes in coal, please let me know. For Franklin, data must dictate the model. In 1947, seeking a change in her research environment, Franklin moved to Paris to work at the Laboratoire Central des Services Chimiques de l'État. Under the direction of Jacques Meringue, she mastered the techniques of X-ray crystallography as applied to amorphous substances. Franklin flourished in the egalitarian and intellectual climate of Le Labo, becoming fluent in French and earning a reputation for her precision and effortless skill in diffraction. In January 1951, she accepted a 3-year Turner and Newall Research Fellowship at King's College, London. The transition proved difficult. King's College was a far more traditional institution than the Parisian lab she had left behind. Franklin was excluded from the senior common room and smoking room, which were reserved exclusively for men. A series of institutional misunderstandings regarding her role led to a breakdown in communication with Maurice Wilkins. While Wilkins assumed she was hired as his assistant, Franklin had been assured by the department head that she would lead the X-ray DNA work independently. Focusing on control of her sample fibers, Franklin identified that the structural state of DNA was a function of its environment. Through meticulous calibration, she and her student Raymond Gosling discovered two distinct forms. The A form was a dry crystalline state existing at roughly 75% relative humidity. The B form was a wet paracrystalline state existing at humidity levels exceeding 75%. Control of the humidity was the only path to clarity. Franklin engineered a specialized chamber for this purpose. She maintained constant moisture levels by bubbling hydrogen through salt solutions into the camera. This environmental stability allowed for exposures lasting as long as 100 hours. Her notes reflect a focus on physical constraints. The phosphates are on the outside so that phosphate-phosphate interhelical bonds are disrupted by water. To Franklin, DNA was not a puzzle to be guessed. It was a structural climate control problem to be solved through experimental induction. In May 1952, Franklin and Gosling captured an exceptionally clear diffraction pattern of the hydrated B form of DNA, cataloged as Photo 51. The image revealed a striking X pattern, a geometric indicator of a helix. Technical analysis of this pattern allowed Franklin to determine specific parameters: a helical pitch of 34 angstroms and an inner base distance of 3.4 angstroms. While Franklin recognized the helical nature of the B form, she remained cautious regarding the A form. She refused to assert a universal helical theory until the data for both forms could be reconciled. On January 30, 1953, Morris Wilkins showed Photo 51 to James Watson without Franklin's knowledge or permission. Watson later recalled, "The instant I saw the picture, my mouth fell open and my pulse began to race. The black cross of reflections which dominated the picture could arise only from a helical structure." At the same time, an internal Medical Research Council report was circulated for a routine review of the King's unit. The document contained Franklin's unpublished measurements, including her identification of the unit cell as having face-centered monoclinic C2 symmetry. This detail was decisive for Crick. It revealed that the two sugar-phosphate backbones must run in opposite, anti-parallel directions. Within weeks, Watson and Crick completed their double helix model using these measurements as their empirical foundation. The discovery of the double helix was published in 3 back-to-back papers in Nature on April 25, 1953. The Watson and Crick paper only vaguely acknowledged being stimulated by a knowledge of the general nature of the King's results. Franklin's data provided the essential verification for the model. Rosalind Franklin died on April 16, 1958, at the age of 37 from ovarian cancer. Her illness was likely influenced by her extensive exposure to X-ray radiation. Safety protocols of the era were insufficient. In 1962, the Nobel Prize for Physiology or Medicine was awarded to Watson, Crick, and Wilkins. The Nobel Foundation statutes strictly prohibited posthumous awards. Additionally, the prize could be shared by no more than 3 individuals. Because Franklin was no longer alive, she was structurally ineligible for consideration. In March 1953, she moved to the Biomolecular Research Laboratory at Birkbeck College. There she shifted her focus to virology and the tobacco mosaic virus. Using isomorphous replacement, adding heavy atoms like lead or mercury to the virus protein, she determined its precise helical geometry. She demonstrated that the virus was not a solid rod, but a hollow tube. Her most significant contribution was locating the viral RNA. She established that the single-strand RNA was embedded deep within the protein subunits at a radius of 40 angstroms. She continued laboratory work despite severe illness. In 1958, she designed a 5-foot-tall molecular model of the virus for the Brussels World's Fair. She died just 1 day before the model was unveiled to the public. The record of Rosalind Franklin is a study in the mechanics of control, from measuring nanometric pores in coal to resolving the anti-parallel strands of DNA and the hollow architecture of viruses. Her work was defined by methodological rigor. She belonged to a tradition where measurement precedes theory. The common narrative of stolen credit often simplifies a more complex institutional reality. Franklin herself viewed science as a collaborative accumulation, reportedly telling Gosling, "We all stand on each other's shoulders." In the end, data outlives the narratives constructed around it. Institutions distribute recognition, but the archive preserves the measurement. Her legacy lies not in a single model, but in the discipline of measurement that made models possible.